The overall goal of this positron emission tomography computed tomography, or PET-CT image analysis procedure, is to study the disease process and potential treatment methods of Mycobacterium tuberculosis infections in non-human primates.
This method can help answer key questions in the study of tuberculosis, including infection location over time, the reactivation from latency, and the outcome of specific treatment regimens.
The main advantage of this technique is that Mycobacterium tuberculosis infection and treatment can be tracked in real time without the need for periodic animal necropsies.
Begin by exporting the co-registered PET-CT images to the appropriate software and opening the images from the software database in the axial orientation. Click on the CT scan and change the window level window width to CT pulmonary. The lung tissue will become dark and the anatomical features will appear lighter, with the airways in black and the vasculature in white. Focusing on one lobe at a time, scroll through the fused PET-CT image to identify the lesions. Small lesions can be distinguished from the vessels by hovering the cursor over the structure in question and scrolling up and down a slice or two. If the structure remains under the cursor, it is a lesion. To measure a lesion of interest, remove the PET signal so that only the CT is visible, and select the Length tool. Scroll until the slice that contains the largest portion of the lesion is visible, and draw a line across the longest length of the lesion. The data included in this region of interest will represent the length in millimeters of the diameter of the lesion.